30,000 genes that are coding for proteins, they make up 2% of the DNA. So we have around like 98% of the DNA is composed of non-coding genes. وهاي non-coding genes, the final product of it is RNA. Okay, but this RNA هو regulates the gene expression of the 2% of the DNA. تمام، يعني ال 98% regulates ال 2% of DNA اللي هم الجينز ال 20 to 30,000 gene expression. So RNA basically مسؤول عن اللي هو ال structural units اللي بتعمل لنا البروتين اللي هم مين في الخلية، شو بنسميهم؟ وين البروتين synthesized ribosomes. Exactly. Okay. ال RNA كمان مسؤول اللي هو مثلا عن ال stability of the messenger RNA that forms, how fast it gets degraded. Okay. ففي عنا species من ال RNA اللي هم ال siRNA وال micro RNA، هدول مسؤولين عن ال regulation of the lifespan of coding messenger RNA. Okay. فعشان هيك شوي الجين ال traditional definition تبعه تغير. لما نحكي genome, what does it mean?
Just the full DNA in a human body or in a cell.
Exactly. Okay. طيب, what's the difference between a diploid and a haploid cell?
A diploid cell has 46 chromosomes, while a haploid cell has 23 chromosomes.
ممتاز. Okay. And how many of these 46 chromosomes are autosomal chromosomes, and how many are sex chromosomes?
44 are autosomal, while 2 are sex chromosomes.
Exactly. Okay. طيب. What do we mean by homologous chromosomes?
They are pair of chromosomes.
الشاب خلينا نعطي فرصة.
replicated chromosome في الديبلويد.
Okay, في ال diploid cell we have اللي هم الكروموسوم homologs اللي هو إيش؟ إنه في عنا one paternal and one maternal chromosome، فهم على شكل pairs عادة. Okay. بس بالهابلويد cells we have إيش؟ half of the chromosomes. فبالتالي السبرمز فيها راح يكون عندي how many؟ والoocytes how many؟ ما راح يكون في عندي homologous chromosomes. راح يكون half of the number of chromosomes. Okay. ولما يصير عندنا fertilization ال fertilized egg راح تكون عندي diploid. ممتاز. لما نحكي allele, what does it mean? كلمة allele.
A gene copy.
Okay, and how many alleles do we have for each gene?
2 alleles.
Okay, 2 alleles. Each one is on إيش? one of the pairs of chromosomes. وهذا الحكي حكينا إنه one allele بيكون على one pair من ال homologous chromosomes، والother allele بيكون على ال other pair ال other chromosome of the homologous pair. طبعا one of them بيكون from the mother side, another one is from the dad's side. Okay. طيب.
دكتورة.
Yes.
هلا في بالرسمة اللي بالسلايدات اللي بالقبل إنه هلا السنجل كروموسوم it has 2 sister chromatids. the single chromosome has 2 alleles. Yes. هلا السنجل chromosome has 2 alleles ولا هو بنعتبره 1 allele؟
No, it's 1 allele لأنه these are exactly the same. Okay. فهم replica of each other. تمام. لكن what we mean by allele اللي هو إنه one allele is carried at one of the like at the paternal chromosome and another one is carried in the maternal chromosome. Okay. فإحنا when we refer to alleles, okay, we refer to those two ones, one in the dark purple and another one is in the light purple. Okay. these are, we consider them different alleles for the same gene, okay? So they are two copies of the same gene, تمام? يعني not the other one, okay? طيب اوكي هلا بالنسبه للدي ان اي طبعا we know انه في عندنا تقريبا في الهابلويد جينوم عندنا around 3 billion base pairs of DNA. فالدايبلويد سيل رح يكون فيها الضعف اللي هم 6 billion. والدي ان اي طبعا هون في بس شويه يعني fun facts about the DNA اللي هو انه اللينث تبع الدي ان اي إنه لو الsun is 150 billion meters from Earth, we have enough DNA to go from here to the sun and back more than 300 times. So this is a very long molecule, okay? So how is it possible for this molecule to be packaged in a small organelle inside the cell called طبعاً اللي هو nucleus? So how is it possible to package it this way? Well, answer to this is that in the cell, we have the DNA packaged as a very compressed chromatin, okay? ورح نحكي more in details about chromatin composition and the types of histones next week when we talk about mitosis and the segregation of chromosomes. But here, this picture illustrates the different levels of DNA packaging. Okay, from 1 you have to 7, and you can see how this long DNA is compressed into these fibers اللي هي بالنهاية بتكون they form the two sister chromatids of one chromosome. Okay, فمثلا at the simplest level you just have the double-stranded DNA, okay? And this double-stranded DNA wraps itself around 2 times, okay? يعني هي 1.65 times ممكن مرة ونص around histones. و this structure we call it nucleosome. And then these nucleosomes, each of them consist of 8 histone proteins. Then the nucleosomes fold up to produce a 30 nanometer fiber, okay? And this fiber forms loops averaging in 300 nanometer in length. Finally, we have a fiber that is 700 nanometer in width. And this fiber, okay, it makes one أو the width of one sister chromatid. زي ما أنتم شايفين إن the width of 2, which is a whole chromosome, is around 1400, which is double the 700. Okay, so these are the levels of packaging which the DNA undergoes in order to fit inside the nucleus. Imagine that every time our DNA replicates, okay, it has to separate أول إشي حتى during gene expression it has to unwrap from around the histones, and the 2 strands need to then separate in order for the DNA to replicate itself for طبعا cell division or in order to make a messenger RNA or any kind of RNA. So in eukaryotes, this process is time-consuming and energy-consuming because the cell needs also to synthesize the same number of histones in the daughter cell in order for the new DNA to be packaged in exactly the same way. ورح نحكي عن the types of chromatin. Based on this packaging, we have regions in the DNA where the chromatin is active and other regions where chromatin is inactive. We have euchromatin and we have heterochromatin. We'll talk about it in details next week. And the tightness of the chromatin actually dictates the fate of gene expression. Okay, so it's a very it plays a very important role in regulation of gene expression. So this slide shows the same idea, except we have electron microscopy images. So we have real images here, in addition to the illustration. Okay, so you can see under the electron microscope how each of these structures looks like. So here we have just a double-stranded stretch of DNA without any histones, and then, you know, for the DNA that already had formed nucleosomes, it looks like beads. Okay, and here at the 30 nanometer fiber, you have like thicker structure already, and finally you have the structure of the final, the width of the sister chromatid. Okay, after the packaging is fully done, and it's represented by the chromosome electron microscopy image here. Okay. Okay, some revision of the components of DNA and RNA and what are the differences between DNA and RNA. So what does DNA stand for? Deoxyribonucleic acid. You already know this, it's written in the slide also. RNA is ribonucleic acid. Okay, and what's the difference between them in terms of structure? As you can see here, if we look closer to the DNA molecule and the RNA molecule, the first difference between them is that the sugar in the DNA is a deoxyribose, but it's in RNA a ribose. Okay, so we have missing oxygen here, okay, compared to RNA where we have the hydroxyl group here. Okay, nucleotides. What does a nucleotide compose of? And what's the difference between nucleotide and nucleoside? Anyone?
Doctora, two nucleosides make a nucleotide?
Actually, no. It's the component of the nucleotides. There are 3 components in the nucleotides and 2 components in the nucleoside. So the nucleotide has the sugar, ribose or deoxyribose, a phosphate molecule, and a nitrogen-containing molecule, which is one of the nitrogen bases that we have in the DNA or the RNA. Okay. A nucleoside, it's only a sugar with a nitrogen-containing molecule without a phosphate molecule. Okay, so here if you see my mouse, so if we remove the phosphate group, okay, from carbon number 5 of the sugar, then this becomes a nucleoside. Okay, it's only a sugar and a nitrogen-containing base. Type. How many nitrogen-containing molecules do we have in the DNA and RNA? And are there any differences? Anyone?
Doctora, 4 for DNA and 4 for RNA, but apparently 1, which is thymine in DNA, while the rest is in RNA.
Mumtaz. Okay, so these are the differences. And how are these nitrogen-containing molecules are connected? Because we have 2 strands of the DNA, and we have, you know, nucleotides on each strand. And so to keep the 2 strands together, there are bonds that are connecting these nitrogen-containing molecules. What kind of bond is that, and how many are there between, for example, adenine and thymine, or cytosine and guanine?
Hydrogen bonds.
Hydrogen bonds. Okay, how many do we have between adenine and thymine?
2.
2. And cytosine and guanine? 3. Okay. So when it comes to the stability of DNA, as you know, our DNA is stable under our body temperature, which is around 37, and it can be fully denatured if you increase the temperature to 95, which happens in vitro when we try to perform PCR reaction. So the stability of DNA in our body is regulated by these hydrogen bonds between the nitrogen bases, but also by what kind of bond, which is stronger than the hydrogen bond.
Phosphodiester bond.
Okay, phosphodiester bond, exactly. So if we look at the DNA structure, so we have the 2 strands running in opposite directions, right? That's why we call the DNA anti-parallel, because we have one strand running from 5' to 3', okay? And another one is running from 3' to 5'. And if you look at the blue, this is the sugar phosphate backbone. So this one is stabilized by the phosphodiester bond that connects carbon number 3 from the previous sugar molecule to carbon number 5 of the next one, okay? Because the growth of DNA, which is polymerization of the DNA, it actually progresses in the 5' to 3' direction. It doesn't progress in the opposite direction, okay? So the addition always and building a new phosphodiester bond should always happen to carbon number 3, okay? Do you know why it can't progress in the opposite direction? Polymerization reaction.
عشان دكتور النيوكليوتايد بيكون عندي 5' end فما بقدر نبلش ما بقدر DNA polymerase to bind to it.
Because ايش ال 5' end عليها؟
النيتروجين بيز اللي هو جوانين او ثايمين او سيتوزين او ادينين.
هلا هو هلا to make things more clear, so the addition or the polymerization is always happening here in this direction. Okay, do you see carbon number 3 in the deoxyribose, and this is carbon number 5 where a phosphate group is added. Okay, so you will have another carbon number 5 coming from the next nucleotide, and a phosphate group will be added connecting, okay, the 3' carbon of the previous nucleotide. And the reason is the presence of this hydroxyl group. Usually hydroxyl group, okay, presence or hydroxyl group usually accepts a phosphate group. For example, the 3 amino acids that we have in the cell that can be phosphorylated, I mean, a phosphate group can be added to them, is they're tyrosine, serine, and threonine. And what's common between these 3 amino acids is the presence of the hydroxyl group that accepts a phosphate group, okay? So the phosphodiester bond formation should happen between the carbon number 3, which has the hydroxyl group. The phosphate group will basically راح تعمل hydrophilic attack للhydroxyl group and the new phosphodiester bond will be formed, okay? This is also true for the 3 amino acids that usually accept phosphate group in our cell. They accept it because they have a hydroxyl group in the side chain, okay? طيب هلا هاي بس الستركتشرز البيورينز والبيرميدينز المكونة لل DNA and RNA والdifferences بينهم just for revision. So we have usually adenine and guanine. We have 2 rings, and cytosine, thymine, and the uracil. In RNA, they have 1 ring. So this is the difference between them. Now, حكينا the stability here is assured by the sugar phosphate backbone زي ما انتم شايفين. So this is what I was talking about. If you compare this, the carbon number 3, it has a hydroxyl group here, and then we have the phosphodiester bond formed between nucleotide number 1. So the hydrogen, okay, was reduced, and then a new phosphate bond formed between carbon number 3 from the previous nucleotide and carbon number 5 from the added nucleotide. Okay, so we have phosphodiester bonds and we have hydrogen bonds that are responsible of the stability of the DNA structure. وحكينا إنه we have 2 hydrogen bonds connecting adenine and thymine, and we have 3 connecting guanine and cytosine. And so it's easier to separate DNA strands in regions that are rich with adenine and thymine. وعشان هيك إنه the origin of replication in our DNA, أو كل origins of replication in our DNA are usually rich with adenine and thymine, because this is where replication is initiated, and it's easier to separate these regions. DNA strands in regions that are rich with adenine and thymine. وعشان هيك إنه the origin of replication in our DNA, أو كل origins of replication in our DNA are usually rich with adenine and thymine, because this is where replication is initiated, and it's easier to separate these regions than regions that are rich with guanine and cytosine. So if you get such a question, basically knowing that there is complementary base pairing between the 2 strands of DNA, this means that a double helix, okay, will have the same composition of guanine and cytosine, and the same composition of adenine and thymine, which means that if I give you the information that a certain DNA molecule is composed of 40% of guanine and cytosine, then you would right away be able to tell me that there's around 21% guanine and 21% cytosine, and then the remaining, which is in this case 58, is divided by 2. You have 29% of adenine and 29% of thymine. Okay, طبعا this is not necessarily true for single-stranded DNA. So this is only true for double-stranded DNA. That's why Watson-Crick rules for base pairing. These rules are meant for a double-stranded DNA. We have viruses, for example. Some of them have double-stranded DNA, some of them have single-stranded DNA, so we cannot apply the same rule on the single-stranded DNA. Okay. طيب حكينا عن الانتي parallel. Okay, we can predict. So the complementary base pairing and the anti-parallel nature of the DNA double helix allow us to not only predict the percentage or the composition of purines and pyrimidines if we were given a percentage of one of these groups, but we can also predict the sequence of the strand. So if you were given the sequence of one of the strands of DNA from 5' to 3', and I asked you to write the complementary sequence, then you would be able to do so, okay? And this is due to the anti-parallel nature of the DNA and the composition of these nitrogen or the type of complementary base pairing between the nitrogen-containing molecules. Now, RNA, as you might know, most RNA species are single-stranded, but when they are actually produced during transcription in our cell, they usually form secondary structures that look like hairpin, okay? This is what we call a hairpin. For example, a tRNA would have such a structure, and this is due to complementary base pairing between nucleotides that are close enough, and they can form complementary base pairs. So you would end up with such structures when we talk about RNA. And this is to benefit the RNA, okay? Because if the RNA is single-stranded, شو راح يصير لو ضلوا ال RNA single-stranded في الخلية؟ حتى ال DNA, if it's a single-stranded nucleic acid, شو ممكن يصير؟
Degraded.
أيوة, degradation, okay? So we have nucleases اللي هم nucleic acid degrading enzymes. في عنا ribonucleases وفي عنا DNases, okay? So DNases, they usually degrade DNA, and RNA nucleases, they usually أو ممكن نسميهم كمان RNases, okay? They degrade RNA. And so the RNA that's produced during transcription forms secondary structures in order to protect itself from nucleases attack, okay? لأنه زي ما حكينا, it plays an important role in regulating the gene expression, okay? And other processes also like post-transcriptional modifications اللي بتصير لل RNA after transcription, okay? So it needs to stay around for some time and protected from nucleases. تمام. هلا إيش السنترال دوغما؟
إنه DNA get transcribed to RNA and then translated to a protein.
Okay, central dogma, yes, it explains the flow of genetic information from DNA to RNA and then to protein for دايما خلينا نركز for gene or protein coding genes, okay? لأنه زي ما حكينا إنه 98% عندنا non-coding genes و 2% عندنا coding genes, تمام؟ فالسنترال دوغما طبعا this flow لو اجينا سألنا بعض الأسئلة, okay, حكينا ايش السنترال دوغما, ايش البروسيس of generating RNA from DNA is called? Transcription, okay. وايش البرودكت of transcription? Is it only messenger RNA? Anyone? How many examples do you know of RNA that is generated by transcription from DNA?
tRNA, rRNA.
Okay, so yeah.
But that's what I can remember, honestly.
Okay, tamam. So we have messenger RNA, which is the RNA product that is usually used in translation to produce proteins or polypeptides. And we have other species of RNA like tRNA that participates in translation of proteins. We have ribosomal RNA that's actually a functional part of the ribosome. And we have small nuclear RNA and small nucleolar RNA, and these play important role in post-transcriptional modifications of other RNA products. We also have siRNA, which is short for short interfering RNA, and we have the microRNA or miRNA, and these two play important role in regulating the lifespan of messenger RNA. Therefore, طبعاً, they regulate the fate of the translated proteins and the quantity of the translated proteins. عرفنا الجين. What's a codon?
It's the coding part of the gene.
Okay, it's a coding part of a gene. So how many nucleotides do we have in a single codon?
3.
3, exactly. Okay, so it's a sequence of 3 nucleotides that codes for a certain amino acid. Okay, so a codon, unless it's like stop codon, but a codon, okay, usually is represented by 3 nucleotides that are identified or translated, خلينا نحكي, to a single amino acid. Okay, طيب. The process of generating polypeptides from RNA is called
translation.
Translation, okay? And why the first step in gene expression is called transcription, and the other step, which is the second one, to produce polypeptides, is called translation?
Doctora, maybe because the first step we are generating a complementary strand, while in the second step we are converting it into a protein in case of coding the gene.
Okay, so where do you have the language of information changing, in transcription or translation?
Translation.
Okay, that's why it's called translation. Transcription, it's like, for example, you know, when 2 people speak Arabic with different dialects, okay, and so one of them will reproduce a copy, okay, of one of those dialects. It's still the same language, okay, but he produced a copy that's maybe clear, easy to read. That's why from DNA to RNA, we're using the same language. It's the same product, basically. We are just copying information. That's why it's a transcript. That's what we call, by the way, every RNA molecule produced from transcription. It's a transcript. And then polypeptides, we are changing the language of nucleotides to the language of amino acids. That's why it's called translation, okay? How about the process by which DNA molecules are multiplied?
Replication.
Replication, okay. Do you know any methods maybe you studied or you were exposed to that can be used to study or manipulate DNA or RNA or proteins?
PCR.
So what do we use PCR for? To study what? DNA, RNA, or protein?
DNA.
DNA. Can we use PCR to study RNA?
I'm not sure, but I don't think so.
Actually, we can. We can use PCR to study RNA. We produce complementary DNA from RNA using PCR. Real-time PCR is used to study the quantity of gene expression, which is represented by how many RNA molecules are produced from a single gene, okay? But يعني, usually the conventional PCR that we have in the lab is used to study DNA, okay? The simple PCR. Now, any methods, you know, that we use to study RNA?
Doctor, maybe gel electrophoresis.
Gel electrophoresis. Okay, so we use it to see what? The RNA bands, right?
Yes.
Okay, tamam. And if I want to identify certain RNA, like I want to identify a certain RNA molecule, what do we use for that?
RNA labeling.
Okay, RNA labeling. Exactly. Do you mean like northern blot?
Pardon?
Do you mean northern blot? Using probes?
I'm not sure about the name, but what I remember is that we can use fluorescent light to mark the strand that we want to test and make sure if it's there or not.
Exactly. Okay, so we call these hybridization techniques. So we can use fluorescently labeled sequences of RNA that are complementary to an RNA of interest or the target RNA that we're looking to identify. And so they basically can hybridize, and we can quantify the signal, and the signal will tell us how much of this RNA is present, and based on the sequence of the probe, because we already know the sequence, we would be able to identify the type of RNA we found, okay? وهذا اسمه في الجينيتيك testing والجين expression أو الترانسكريبتوم analysis مايكرو arrays، مش مايكرو RNA اللي هو حكيت عنه one of the RNA species. لا، هي technique called micro array. It's based on hybridization between a probe that we design and the target RNA we're looking to identify. Okay, تمام. Any methods to study protein? Identify protein, quantify protein.
I remember there was a mechanical way where we suspend the proteins using their complements in a liquid. I don't remember its name, but you can it's a quantifying procedure to know the amount of proteins present in a liquid.
Yeah, liquid chromatography, you mean?
Yes.
Yes, liquid chromatography is one of them. We have also Western blot that is also used to identify candidate proteins we're interested in studying, especially يعني الناس اللي interested in molecular biology. So this is one of the main techniques that we use in the lab, okay? And these techniques, it depends on the scale of identification we're looking for. If we are interested in identifying a few candidate proteins, there are small scale methods for that. But if we're interested in identifying the whole proteome of a cell or organism, then we go to what we call proteomics, okay? You will get used to this. So genome, transcriptome, proteome. Genome حكينا إنه اللي هو entire gene composition أو genetic composition in a cell or organism. Transcriptome اللي هو entire RNA. وproteome هو entire protein population in a cell or in an organism. And there are small-scale methods and large-scale methods to identify these molecules, okay? طيب. Now, we have to differentiate also. I'll see if you could differentiate between a genotype and a phenotype. Okay, so what do we mean by a genotype?
It is, what does the gene code for in terms of genetics, not in terms any phenotype. It is what the gene manifests as in the human or in the organism. Like in the
Exactly.
genotype, it's only the name of the gene in terms of genetics. Yes.
Okay, so a genotype is basically the genetic sequence or composition in regards to a particular trait. Okay, phenotype is the trait itself or the manifestation of that genotype. Okay, so for example, we might have a gene that's responsible of producing brown eye color. So if this gene is represented by two alleles, and we say like it's let's say AA, okay, so we're talking about a genotype. The color itself is a phenotype. Tamam? Now, genes are responsible not only for just the phenotypes that we see in a population, but actually every molecular and cellular mechanism or process in an organism is controlled by orders from different genes. So we have genes that are producing different molecules. These molecules, some of them function at the cell membrane, some of them function in the cytoplasm, some of them are part of the cytoskeleton, okay? And these molecules, they interact through signaling pathways and communications in order to produce a certain phenotype for a cell, okay? And they're responsible for what we call cell differentiation, because if we take all our cells and we look at the DNA component, you'd find that it's all very similar. Okay, there are no differences in the DNA component of epithelial cell or endothelial cell. But the phenotype of these cells is different. And the reason is because each of these cells activates a very specific pattern of gene expression. So there are certain genes that are on in epithelial cells and off in endothelial cells. And there are genes that are on in endothelial cells but off in epithelial cells. And that's what we call cell differentiation. So cell differentiation is a cellular process, but it's defined at the molecular level by differences in patterns of gene expression. Okay. وهي اسمه بالعربي اللي هو التمايز الخلايا. Okay, they become specialized. They produce different functions نتيجة لأنه they have different patterns of gene expression. Now, another concept or principle that's important in genetics is the effect of the environment. Okay, تعرفوا إيش الدifference between genetic and epigenetic? We have genetic changes or genetic variations, and we have epigenetic changes or epigenetic variations. Anyone?
دكتورة، الجيناتيك هي اللي بتكون الصفات اللي بتحكم فيها الجينات بس لحالها، والإيبيجيناتيك بتكون عليها العوامل البيئية والمحيطة هي بتأثر على التغيرات هذه أكثر من الجينات.
Okay, ممتاز. So genetics أو genetic changes هي عبارة عن changes in the DNA. Okay. Epigenetic changes هي changes outside of the DNA. طبعا عادة هي بتكون in response to environmental factors when it comes to our cells الابيجينetics المسؤول عنها الإشي اللي أنا حكيته لما حكينا عن الكروماتين أوكي اللي هي for example chromatin activation inactivation of the chromatin اللي هي how tightly the chromatin is packaged or how loosely the chromatin is packaged dictates the fate of gene expression. So now we're talking about changes outside of the DNA, but they actually affect gene expression. Another example اللي هو DNA methylation. DNA methylation is also something that regulates gene expression, but it's actually not dictated by the DNA sequence. And the effect of the environment is usually it also explains the epigenetic phenomena that we have. That's why في عنا طبعا epigenetic changes ما بتكون inherited لأنها it's outside of the DNA. ممكن تصير بالسيل نتيجة ل response to a certain stimulus okay ممكن lifestyle معين الإنسان بيكون عايشه والenvironmental factors اللي هو exposed to it can affect how his genes or her genes are functioning okay لكن هي عادة ما بتكون inherited ف this is an important concept because many disorders that have genetic components, they actually are classified under multifactorial disorders, which means that there are genes that act as risk factors for these disorders, but also environmental factors play a very important DM. Type 2 DM, okay, genetic or geneticist identified what we call genetic risk factors, which means certain genes or genotypes or polymorphisms ممكن تكون common among people who share this disorder, but the environmental factor plays the most important part in the pathogenesis of type 2 DM. Okay, so this is an example of on one فيها environmental factor plays an important role. Okay, لكن we have genetic disorders where it's very stable that this mutation, if it's found in a certain gene, then this mutation will produce a genetic disorder. And this is true for most of the single gene Mendelian genetic disorders. طبعا, as you know, the information in our DNA generates a huge diversity. And we're talking about 4 nucleotides that are repeated along a stretch of DNA, but we have this diversity in our world when it comes to the animal kingdom and the plant kingdom. طبعا, there is a field of genetics that is interested in studying the DNA of the different organisms. We call it comparative genomics. So they basically try to find the percentage of homology between the different animals or the different species. For example, most of the clinical studies that are related to human disorders are carried out in mice or mouse models because there is a very big homology between the DNA of a mouse and the DNA of a human. And as you can see here, for example, if we took cytochrome C protein as an example, إذا بتتذكروا cytochrome C, it's one of the important proteins in the mitochondria, plays a role in oxidative phosphorylation and the electron transport chain to produce energy. If we actually look at the amino acid sequence of cytochrome C and compare it across of different species, you would find great homology between the amino acids. And this is actually identifying the homology between the different species helped scientists to study many proteins that are of importance to human and play important role in development of different diseases. Knowing the sequence of these proteins allowed us to actually manipulate the sequence, introduce mutations in the sequence artificially, and see what's the consequence of this mutation, okay? So this is basically what a molecular geneticist would be interested in knowing. And also, these sequences are all deposited in the database. So if we discover a new organism, for example, a very recent thing that happened in the world, which is COVID-19, right away after sequencing the genome of the COVID-19, scientists were able to tell that it belongs to the family of coronaviruses. How did they know that? They basically, after sequencing the genome, which means they identified the sequence of nucleotides of the whole genome of this virus, and they compared it to the genomes deposited for other viruses in the database and found the percentage of homology between them. So there are domains that were conserved among the viruses that belong to the family of coronaviruses, okay? So this is an important thing to know in genetics. Also, knowing the sequence of different genes basically allow us to understand or to study the consequences of the variations or the mutations or the polymorphisms that can happen, and then we could study the frequency of these polymorphisms among a group of patients with a common disease, okay? Or study even the percentage or the frequency of this polymorphism in a whole population of Jordan. Definitely knowing these sequences is of importance to any geneticist. The fun thing about genetics is that you can play.
أعتذر عن المقاطعة بس لأنه في عنا هلا محاضرة ثانية.
Okay. I'm so sorry. You can go ahead to your next class. كلكم? Everyone?
هي مشتركة.
Okay. It's fine. I'll record the rest of it and deposit it in files.
تمام دكتورة، يعطيك العافية.
Okay. Bye.
مع السلامة.